So as Professor Shankla introduced me, I'm Shankla, I'm in the Executive Lab, and I'll be discussing recent insights into a promising new next-generation sequencing technology called nanopore-based DNA sequencing. So a little bit of practical applications of DNA sequencing. In a clinical setting, a physician may tailor some drug therapies towards their patients, to mitigate any sort of side effects from various drugs. So that's sort of a futuristic sort of scenario. In a more practical scenario, that would be more familiar to you and I. In the lab, instead of waiting for days for sequencing results, we may be able to get sequencing results in real time. So this is the mid-ion nanopore-based DNA sequencer by Oxford Nanopore. And they claim to do real-time DNA sequencing. Now it's currently being tested. But my point here is that inexpensive and rapid DNA sequencing can really bolster instrumentation in the physical sciences. So our group, in collaboration with Oxford Nanopore, have been investigating graphene as a membrane material for nanopore-based DNA sequencing. So before I jump too far ahead into this, let me give a basic introduction into what nanopore-based DNA sequencing is. The physics of the setup is relatively simple. We have two compartments, each filled with an electrolyte solution, positive and negative ions. Now, separating the compartments is a membrane, and embedded in the membrane is a tiny hole called a nanopore. So we place DNA, this electronegatively charged molecule, into one of the compartments, applying an electric field. DNA eventually finds the nanopore and begins to translocate through the nanopore. So now, as the bases are translocating through the nanopore and affect the nanopore, the bases inside the nanopore affect the ionic current to varying extents. So in principle, we may be able to read out the bases inside of the pore from the ionic current measurements. So I study graphene. So I'm studying graphene, which is this atomically thin material, is TEM image graphene, for nanopore graphene, for what you call. This atomically thin material with a high in-plane conductivity. Now, I built a computational structure of this. Here's a single-stranded DNA in a sheet of graphene. Add water ions, perform the simulation using MD methods, where atoms move according to classical equations of motion, and obtain a trajectory of here's DNA on graphene. So in our simulations, we found that DNA translocates through graphene in a stepwise manner. So in this movie right here, we see that the bases of DNA, which are hydrophobically adhered to the graphene, are translocating through graphene under an electric field. So as one base at the edge of the nanopore goes into the nanopore, it dislocates the base currently in the nanopore, which then binds to the other side. So we see this nice stepwise behavior. However, the duration of the steps is stochastic, which may introduce errors in the base readout. So we really wanted to control the translocation of DNA through these nanopores. So we decided to take advantage of graphene's high in-plane conductivity to apply a local potential on the membrane, which we model as charge in simulation. To be able to control the DNA, so since DNA is this electronegative molecule, we could apply a negative charge to the membrane, which would unbind the DNA from the membrane and allow it to translocate through. And if we were to apply a positive charge to the membrane, it would really bind the DNA and hold it in place with that translocation. So we really wanted to reduce the stochasticity in these steps by using this method. So let's take a closer look at DNA on charge-neutral graphene. So here we have this DNA base. This is poly-BT20. It's a homopolymer of 90. Each of these bases are parallel and flat on graphene. And there's parallel flat combination. Now, when we apply a negative charge negative 2 over 10 squared, we see that there's repulsion between the electronegative phosphate groups in the backbone of DNA and the negatively charged graphene membrane. As you can see in this movie here, here's graphene on this charge-neutral membrane, and then we apply a charge of -2, and it unbinds the membrane. So that's what we expected. Now, what happens when we put a positive charge on the graphene membrane? We see something interesting. The bases tilt, and they tilt due to their individual dipole moments, and continuously tilted bases stack. And we can see that here, where we have this DNA in this flat conformation on graphene. Now we apply the charge of -2 onto graphene. And then we can see there's three sort of a triplet of stacked bases right here. So that was only poly-DT. What about homopolymers what about other homopolymers? So we can see that different homopolymers have different amounts of flat, tilted, and unbound bases. So we can take a look at purines like guanine on positively charged graphene. We see that there's some flat bases of guanine and some unbound bases where the phosphate group is bound very tightly, displacing the base. However, with adenine, we see a greater amount of unbound bases and less flat bases. So then we can also take a look at cytosine and methylated cytosine, where the methylation is epigenetic modification. And we see that with cytosine, there are some of very few tilted bases. However, with methylated cytosine, there are many more bases in these tilted conformations. So we also see one really cool and interesting conformation, and that's this upright conformation, where the DNA backbone is not in contact with a negatively charged graphene sheets that occurs at negative charge, and this is guanine. But the amine groups of guanine are in contact with the membrane. So what we have here is an interplay between electrostatics, charge screen, and hydrophobic interactions of the DNA strand and graphene. And through this, we really get complex and rich physics which emerge to create this sort of molecular gymnastics, is what we like to call it. So going back to our original question, we really wanted to know how charge on the membrane would affect the translocation of DNA through the membrane. So applying a transmembrane bias, we saw that translocation on a negatively charged membrane did not occur, which was contrary to what we initially thought. So we were fairly surprised. And on the positively charged membrane, we saw that DNA translocated right away. And we saw this over through multiple simulations and multiple sequences. So we really wanted to adapt this idea of applying charge on the membrane to controlling the translocation and reducing the stochasticity of basic DNA transport through a membrane. So we do this through this concept of stop-and-go DNA transport, where we have a finite amount of DNA translocating through a membrane. This is a positively charged membrane we allow our DNA to go through under this half-whole bias. Now we're about to run out of DNA, so we stop translocation by applying the negative charge to the membrane. Now we're about to run out of DNA, so we flip the direction of the electric field to force DNA to move in the opposite direction, and then we can allow it to translocate. So really, in the end, we observed two things in this study. We see that really complex and rich physics sort of emerges from this very simple system of DNA on charged graphene. And the second, and I think more important point is we can really reduce the stochasticity in the translocation of DNA through an nanopore. So hopefully, I'm hoping this technology can be effective in driving sequencing past the sub-1K level in the future. And I'm very quickly want to acknowledge my advisor, Professor Xavier, and various funding agencies, including the CBLC, for allowing me to run this talk. Any questions?
Questions from the audience?
When you start the simulation, you already have the DNA through the graphene?
Yeah, it's pretty threaded.
And how, in practice, going to do the first thread through the graphene?
In actual experiment?
Yes.
So from what I understand, in the experiment, they put the DNA in one side and then they apply a field. But now with graphene, it really hydrophobically sticks to the edges. So that's one of the areas that they're currently working on, and we're currently working on how to bring the DNA to the pore. So it's not a very intuitive step.
Right. They may not try to mechanically or something like pulling with it. I don't know. I was just curious.
So there are different ways of doing it. I've heard with protein, using this for protein sequencing. I've seen that they can put like some sort of clip-X protein underneath, which pulls the DNA, which pulls the protein through.
Right, but protein doesn't. Right now you do sequence, so when it's that it's more control.
Right.
Yeah, sure. Let me push back on this a little bit. I mean, as far as I know, that the current Oxford nanopore device has a protein in there that basically controls translocation. It's not just the DNA threading through the pore, right? So, I mean, how realistic is it to just have a sort of hole? I mean, even if it's in a graphene sheet, or could you combine the graphene with the protein to sequence DNA?
Right. So those are sort of three separate areas. Oxford nanopore's their current. I don't remember which specific protein they use in the pore. May either be iPoLS or MSPA, but they can do that. They can embed it in solid-state membranes, but another thing they're looking at is just purely solid-state membranes. From what I understand, in all three fields, we're kind of at an equivalent point of how can we make it better. So it's just sort of three parallel sequences that are working to find the best sort of solution in the end. Hopefully that's a good answer.
Okay, one last question. So, if you're doing sequences, right, what do you do about poly T's, poly G's? How do you distinguish one T from two sequential or three sequential T's, for example?
So that would be the current issue. There are various ways of base calling. It's just not an area I'm an expert in. But if you were to have, so I think this is why the stepwise transport and reducing, keep like sort of a fixed duration of steps is important to be able to discriminate between multiple.
Right. But if they're still somewhat stochastic, then you won't be able to distinguish if it's 2, 3, or 4.
Exactly. But there are methods of base calling that are being developed, especially using AI algorithms or whatever. But that's not an area we're familiar with.
Okay, let's thank the speaker again.